Chapter 14, Genetics. Actually, this is the first of several chapters on genetics, starting with Gregor Mendel and the gene idea. Gregor Mendel, in the mid-1800s, did experiments with breeding garden peas. He was a monk. He's considered the father of genetics. His work is the foundation of what we know about heredity today. Now, the geneticists at the time, the people who studied heredity or at least speculated on it, did not embrace Mendel's idea. Mendel's idea of heredity really wasn't discovered until rediscovered until 1900, so he never saw his own success. Now, with the garden peas, if you look carefully on page 265, table 14.1, he looked at several traits, a number of traits in garden peas. He looked at flower color, in which case he saw two different forms, purple and white. Flower position, axial versus terminal. Seed color, yellow versus green. Seed shape, round versus wrinkled. Pod shape, inflated versus constricted. Pod color, green versus yellow. And stem length, tall and dwarf. So he noticed the same inheritance pattern with all 7 of these traits in these alternative forms. Now, from his breeding experiments of garden peas, he came up with 2 principles, the principle of segregation and the principle of independent assortment. Now, we'll use the example of flower color to discuss Mendel's law or principle of segregation. He noticed that with the purple and the white, he didn't see a blending effect. It was either one color or it was the other. Now, in figure 14.3, he crossed a purple flower plant with a white flower plant, and all of the offspring had purple flowers. Okay? Then he crossed 2 of those, F1s, to get an F2 generation, in which case he got purple and white again in roughly a 3-to-1 ratio. So he saw that whatever caused white flowers didn't go away in that middle generation. It skipped a generation. Nor did he see a blending effect with the purple and the white. Now, he did the same thing with the other 6 traits. He found the exact same inheritance pattern, came up with the law of segregation. Now, the law of segregation basically is that alternative forms of genes exist. Those alternative forms are called alleles. Okay? So for each trait or gene locus, there are alleles. Now, he saw that there are 2 alleles. One of them is dominant, one of them is recessive. In his case, the purple flower allele was dominant. So to use modern lingo for this example, we have the gene locus for flower color, and we have alternative forms, purple and white. So big P would represent the dominant allele, little p would represent the recessive allele for white. Okay? Now, each individual plant would have a pair of alleles, one inherited from each parent. And when the plant was reproducing, making gametes, the letters or alleles would separate. Now, this can be shown on a Punnett square. Now, if he's crossing a purple flower plant with a white flower plant, okay? Now, you got to separate the letters. I'm going to put one parent here, big P, one parent here, little p. Got to separate the letters because that's what happens with meiosis. Now again, during Mendel's time, nobody knew anything about meiosis. Big P here, big P here. Likewise, you separate the letters, little p. Now, what's inside the squares represents the gametes fertilizing into the offspring. These are the offspring. So, big P, bring it down, bring it across. Bring it down, bring it across, down, across, down, across. These are the F1s. They're all big P, and they all have purple flowers because of the dominant gene. Okay? Now, when he crossed those 2, he got another result, and I got to show you another Punnett square. This time he's crossing big P times big P. Okay? Crossing the 2 F1s to get the F2 generation. Got to separate the letters from each parent. A big P goes here, a little p goes here. A big P and a little p. Bring it down, bring it across, big P, down and across, big P, little p. Likewise here, but over here, little p. Purple flowers, purple flowers, purple flowers, white flowers. That's the 3-to-1 ratio he saw in his F2 generation. Okay? So he figured all this out by this law of segregation that we now call it. Now, we need to cover some terminology here. Now, your notes have this example, and your notes have some vocabulary. You'll see the word genotype. The genotype is the genetic description of the organism. Okay? So, these are genotypes. Now, if both alleles are the same, we call this homozygous. So, if both alleles are homozygous for the dominant allele, we call this homozygous dominant. If you got one of each, we call it heterozygous. And if an organism has 2 recessive alleles, we call this homozygous recessive. So, homozygous dominant, heterozygous, homozygous recessive, those are genotypes. But we don't see genotypes, do we? We see traits. We see phenotypes. So phenotype is the expressed trait. In this case, purple flowers or white flowers. Okay? I'll stop this and then I'll continue in the next video.